Hi, I'm Art Mellor. I'm the founder of the Accelerated Cure Project for Multiple Sclerosis, and I, along with Ben Greenberg, are going to give you an overview of what our project is doing and how it relates to transverse myelitis, NMO, and ADEM. Our organization, our mission is to cure MS by determining its causes. We believe that knowing the cause of a disease is one of the fundamental questions that needs to be answered. It allows a number of things to happen. Obviously, if you know what causes a disease, it gives you a lot of ideas of how you might address treating or curing that disease. At a minimum, it allows you to diagnose the disease quickly and accurately. One of the problems in developing treatments for diseases is the lack of good animal models, which, especially in a disease that progresses slowly, generally, like MS, which progresses over years, it's hard to test in humans because it takes so long to find out. It's not like a toe fungus drug where you can, in 12 weeks, know whether it worked or not. You have to spend several years, which makes it very expensive, which makes it unattractive for people to do. So if we could have animal models that were accurate, we could develop drugs more rapidly. These diseases are probably not single diseases. And what causes my MS or somebody else's MS or your TM or somebody else's TM might be different in different people. And therefore, if we can know what the underlying causes are, we may find that specific treatments are more effective for one group of people than for another, and you can get put on the correct treatment more easily if we understood that. Secondly, if we can stop the underlying disease process that's causing it, that can enable repair strategies to work better. That we could, by stopping what's causing the damage, maybe your own body's natural repair mechanisms or some induced repair mechanisms could work better. Because if you can't, the analogy I always use, if somebody's coming and spray painting graffiti on your wall, you might be able to repaint over it, but if they're coming back every night and spray painting on it, eventually you're going to run out of paint. So what you want to do is be able to stop the graffiti artists from coming to the wall at all. And then finally, you may be able to prevent the disease in others going forward. So who is the Accelerated Cure Project? Well, we're a small nonprofit. We were founded in 2001 by me. You can see that I was going to be a nerd from the beginning. I was in high-tech engineer, and I got diagnosed in 2000. And being an engineer, my reaction was, gee, I got this problem. I got to fix it. What causes the problem? And then I was stunned to find that we really had absolutely no idea what causes it. And so in talking with my neurologists at the time, what might get done, and we came up with this idea of making an organization that's focused on determining the causes. We're based in the Boston area. We have 8 full-time staff. And Ben's part of the talk. He'll talk about more about the larger project that involves partnerships and contractual relationships with lots of other people, so it's a relatively large effort. But the most important partnership for today's talk is the partnership we formed with the Transverse Myelitis Association. Thanks to Ben, who made an introduction to Sandy, which resulted in many very long conversations talking about how what we're doing could be beneficial to transverse myelitis and vice versa. And he convinced us that adding into the mix of not only looking at MS, but looking at transverse myelitis, NMO, ON, ADEM, and eventually, I don't list up here, clinically isolated syndrome, which is people with a first demyelinating event, was going to be important in 2 ways. One, any work we do looking at MS, if we can compare the results to these other diseases, we could start to tease apart things that were specific to each of the diseases. And that has a benefit for MS as well as for these other diseases. Now, each of these other diseases at one time or another were all kind of lumped under the same umbrella, generally called MS. And as differences were identified, they were split off and named. And I actually had a neurologist once joke, well, half joke, that the way MS will be cured is we will identify every subtype of the disease, name it something else, that's too small to bother having to research or cure on its own, and then there will be no more MS. And you can see that happening already. So the idea is, can we bring these diseases back into the fold and consider it one pool of diseases that are similar enough that they should be looked at together? And so through discussions with Sandy, we decided that we would change our focus to not just be MS, but to include these other rare diseases, and in addition, to include pediatric subjects. MS is largely an adult disease. It does affect children, but at a very low rate. But these other diseases are more likely to hit in children than MS is. So it became important for us to modify the study that we were doing to be able to enroll pediatric subjects, and we did that too. And as I mentioned, each disease can kind of be a control for the other. So if somebody's just interested in NMO, they're going to have some interest in looking at these other diseases at the same time so that they can compare results. Because if you find a result and say, hey, I figured out something about, NMO, and it turns out what you really figured out was something about all of these diseases lumped together, it's kind of less important. It's not unimportant, but it's less important. The Transverse Myelitis Association has provided us a very generous grant to help fund collecting of the TM subjects. So we have a separate account that we keep, and as we enroll people, like the people that were here today, yesterday, getting their blood drawn, the funding for that will come out of that grant. And for the rest of this talk, because I've kind of patched it together from other talks I've given, where you see MS, think MS and other demyelinating diseases. It's just a tongue twister to say that every time. So everything I'm going to talk about here pretty much applies to all of the diseases together. And if it doesn't, I'll try and point that out. But I don't think there's a case where that's there. So let's just take a look at why we took the approach we did. MS, along with these other demyelinating diseases and many other diseases that we don't know the causes of, has two characteristics that make it difficult to address through standard research infrastructure. One is that the disease does not seem to have a single underlying cause. There doesn't seem to be a gene that gives you the disease, a virus, a toxic agent, a nutritional deficit. But that there's some set of genes that make you susceptible, and then something in the environment that triggers it. So it's a multifactorial disease. But unfortunately, most research is done in just one very narrow area. So if you're looking for the cause of a disease that's got 5 genes and one of 3 viruses that might trigger it, well, there's nobody who really does that kind of research. There's geneticists and there's virologists, and they don't overlap in general a whole lot. So that makes it very difficult for kind of the way research is typically done to uncover how a disease like this is caused. Second of all, as I mentioned before, each of these diseases is probably a small family of diseases, maybe even a large family of diseases, but it's a constellation of diseases. And therefore, when you do a study with 100 people with MS or 100 people with TM, you don't know if you have 4 or 5 different diseases in that pool. And therefore, if you're looking for cause, each subset of person who has the same subset disease is very small. So we need very large, in order to address that, we need very large populations of people with these diseases so we can subdivide them into meaningful buckets as we learn more. And second of all, we need to be looking at the disease from many different angles all at the same time. And these are both things that are difficult to do with the way things are currently set up in the research world. By having a very large-scale shared repository that's run by an independent entity that isn't interested in just one of the different areas, we can address both of these issues. First of all, we can collect very large numbers of subjects together because we can work with many different centers that maybe aren't best friends with each other, but they can each be friends with us. And second of all, we can do the collection in a fashion that is applicable to people who want to look at different things. So we're not just interested in genetics. So there's some big genetics collections out there, and the people are generally pretty good about sharing them too. But they only collected enough data and samples so that you can do genetic studies. If you wanted to come along and do a big antibody study or something, you couldn't do it because they weren't worried about that, and why would they spend their money on research that they were never going to do? So it has to be somebody who's kind of disinterested in each specific, well, is uniformly interested in all the different ways. And the idea being that if we give the geneticists access to these samples and we give the toxicologists and the virologists and the nutritionists all access to the same samples and require that they return their data back to the repository when they're done. They can file their intellectual property and publication rights, but when they're done, they give the data back. Now we can have data from all these different areas, but on the exact same people, so we can now put them together. Because when a geneticist does a study on these people and a virologist does a study on those people, it's very hard to combine the results of those two to come up with any sort of genes plus virus theory, because they looked at totally different people. So by having this shared population, we win. Why can't it be done currently? Well, there's 3 reasons, that any one of which will stop it from happening. All 3 together pretty much guarantees it's not going to happen. One is it's relatively expensive. So, for example, to collect our first 1,000 subjects cost over $2 million. Most researchers will never see a single grant of $2 million. It's rare. So it's an expense level that it isn't insurmountable. I mean, a nonprofit like us, we raise this money from people, and we're able to pay for it. But researchers, it's very hard to do. Second of all, it's administratively difficult. We have, like 8 people working full-time just running the management part of all of this, keeping 6 centers up and running, dealing with all the regulatory paperwork, raising the money, doing all this stuff. If you're a researcher who's doing research, seeing patients, teaching classes, writing grants, and managing grad students, you're going to add on the management of 6 sites on top of that to do a big study? Not going to happen. And then finally, creating this resource is not a publishable act in general. It's not going to advance your career by saying, hey, I just spent 2 years collecting all these samples. Aren't I great? No, you need to be using the samples to get your publications. So as a nonprofit, we can raise the money, we can do the administration, and we don't care about the publication. And then we can make the resource available to the researchers who know what to do with it. So why is it good? First of all, there's a leveraging effect in the number of samples. We bring a bunch of sites together. Right now it's 6 sites. Or let's use 5 sites because it makes the math easier. If each site collects 200 samples and puts them into a shared pool, then people can have access to 1,000 samples by only doing the work of collecting 200. Well, only. It's a lot of work. But you get a leveraging effect in how much is available for everybody. Second of all, I mentioned this a bit before. In the current paradigm, what you do is you have each researcher works on their own group of people and produces their own results that have to do with those people. But if each of these researchers worked on a common pool, that's not only bigger, but it's the same pool that everybody works on, not only do you get the results that they could have anyway, that they would have gotten working on their own, but now you can start combining the results together more easily, and you get more knowledge out of the end by doing this. None of this is like real visionary. It's kind of obvious, but those first 3 reasons I gave is why people aren't doing it. So what we're trying to do is we're creating the largest open access repository. The point is to enable very large-scale multidisciplinary studies to happen, to looking into the causes, subtypes, biomarkers of MS, transverse myelitis, NMO. We want to find out what's underlying cause of this disease. In this year, our goal is to hit 1,000 case subjects for a total. It's going to be a total of 1,500 to 1,800 people in the repository total, because we not only enroll people with one of these diseases, but we will also enroll controls, which Ben will talk a little bit about. So over the long term, what we intend to do is to grow the enrollment. Obviously, there's no upper limit to what we need, because these are consumable resources. You always need more. We can always do bigger studies, and there's always more people who want more. So even if somebody came along and started doing exactly what we're doing, it would be a good thing. We need more. And we provide 2 things. There's a static resource, which is the samples that we've collected and we make available to researchers. And then there's the infrastructure we've developed with these centers that are able to do these collections. We've got very broad consents. We're able to change what we're collecting on the fly without having to go reconsent everybody. And that means that if somebody comes along with an idea saying, hey, you know what? I really need these very specific samples. Like Ben's helping us on a project with NASA where they needed 20 really, really unique samples. It took us about a year to find these 20 people. But we were able to just insert that into the process. We didn't have the samples sitting there, but we had the infrastructure to collect these samples. And so we're willing to work with people to do that.
Now, for the same group of people who saw the questionnaire that you were given to fill out, and you looked at it and you said, these people are insane. I don't remember where I lived 20 years ago. Was I near a coal power plant? I got no clue. It's the same people who've stopped me in the hallway 50 times saying, you know, I was stung by this funny wasp 20 years ago. Could that have had anything to do with it? I don't know. You got to fill out the questionnaire to find out. So the reason these studies are hard and the reason there's so much involved is we don't know necessarily the answer. And to be quite honest, while this is a gargantuan task, I mean a huge task on ART's part, the Accelerated Cure Project's part, and the site's part, it is just as big, if not a bigger task for you. This does not get done without you. This is not about physicians and scientists providing you with some sort of knowledge. This is us learning from you. So whatever you put in, you get out tenfold. So the group of 6 did a spectacular job yesterday. In particular, Jana Goins, who if you haven't met, you should go by and say hi, is our study coordinator and our program director at Johns Hopkins. So Jana, we stole after her degree in public health from Johns Hopkins Public Health. She's an individual who you're going to see out there in the years to come. So did she come to me as a crusader for MS or transverse myelitis? The secret's out of the bag, Jana. No. Jana is passionate about changing healthcare for the masses, a very public health approach to things. She agreed to work with us because she has a unique skill set for organizing and developing these types of programs, and we're hoping to keep her for as long as humanly possible. She made yesterday possible. So think about, there was a centrifuge in the corner. Well, somebody's got to get a centrifuge and ship it out here. We had to find a random place to get 100 pounds of dry ice. You got to ship all these samples. Each of the things you filled out, we fill out in triplicate. All of that's coordinated by Jana and her team, and they did an outstanding job yesterday. So while my... So it's great. I get to get up here and talk about all the success we have, and they're doing all the work. So this is like a dream come true. So there are 6 sites in the United States currently. Number 7's coming online. The MS Research Center of New York, UMass, the Shepherd Center in Atlanta, Johns Hopkins in Baltimore, UT Southwestern in Dallas, Barrows Neurologic Institute in Phoenix, and then in Colorado, there's looks like going to be a new site coming online. 6 sites. Think about the team it took yesterday, just for 40 to 50 people. We've got teams in each of these sites. This is a big, big deal, because in order to do this right, it takes a lot of resources. This is kind of a listing when you get a sense of the effort. You've got the collection sites with staff members at each place. You've got a clinical research oversight group managing the data. You've got the group that manages the actual samples. And you've got a group that just deals with the paperwork and the data and making sure we're in compliance and all of those things. And then the Accelerated Cure Project on top of all that. We have had success. So as of May of 2008, we've enrolled over 1,200 subjects. Okay? It's taking all of this to get to 1,200. 1,200 for one of these studies is a small number. If I'm going to figure out, or if Doug's going to figure out, or if Jana's going to figure out that it's this genetic predisposition with this exposure in that time frame, maybe it has to happen between the ages of 16 and 17 during a full moon and the summer solstice. If we're going to figure that out, the numbers you're talking about to do this are tens of thousands. And it's taking this to get to 1,200. We could never do this at Johns Hopkins alone. We could never do this at Southwestern alone. When I went to my supervisor, my boss, when I was in the initial talks with Art about doing this at Hopkins, he said, it sounds great, but you're committing career suicide. You can spend all this time collecting all those samples. If you don't have a publication to show for it at the end, you just wasted a career. It's a waste of your time. Be very careful. They did not forbid me from doing that, and that was their mistake. So, but they're right. This task does not get done without a third-party entity, and that's what the Accelerated Cure Project brings to the table, as well as a variety of other things, including passion about the project. When we look at the samples that have been collected, obviously there have been a lot of multiple sclerosis samples. It's a more common disorder, and the MS population is very galvanized around this idea. We have been augmenting our transverse myelitis, our NMO, and our ADEM, but we want even more. So as of this date, which was in July, you see TM75, Well, we just added 40 to 50 yesterday. So we came close to doubling the bank for these rare disorders just in this day. So that was a great achievement. And we start to talk about now doing studies. What do we collect? This is technical, but it gives you the sense that we are causation or biology agnostic. I'm not just a virus guy or a nutrition guy or a toxicology guy. I'm not just collecting one set of samples. I'm trying to equip specialists who are interested in DNA to have their DNA, even if I could care less about DNA. The people who want their RNA are going to get their RNA. We want to make sure we capture all of that. You guys know, or several of you know, that case report form you have to fill out is a bear. It is a pain. It is even more of a pain to get all that data into our electronic system, but it's the key. I could draw everybody's blood in here right now, and I should just throw it in the trash unless I know something about you. And the more I know about you is the more I know about that sample, which means the sample becomes more valuable. Okay? The clinical data is almost more valuable than the sample. You can't have one or the other. You have to have both. So the inclusion criteria, we won't go through these. You have to be diagnosed with one of these disorders. You have to be able to give consent. You have to be able to give blood. If you've had certain things that would confuse the picture, and we've had this problem. So, for example, we get a question about bone marrow transplants. If you've had a bone marrow transplant, your DNA is now different. You're a chimera. You have the DNA of the donor floating within you, so we can't sort out certain genetic studies. So there are some strange exclusion criteria here to make sure that we enable the researchers to do the studies they want to do and that they're scientifically valid. The control recruitment, we like to get both parents, if they're available, again, to do genetic studies to try and sort out what genes came from which parent and what made you be at risk. If you can get siblings, adult children, half-siblings, and then the matched unrelated controls. So if your spouse is willing to roll up their sleeve, or if you're willing to threaten your spouse to get them to roll up your sleeve, we will welcome them with open arms into the repository. Guilt goes a long way, by the way. I'm rolling up my sleeve. Don't you care? So what's the researcher request format? So we get phone calls, we get inquiries, people hear about this, they see it on the web, the word's getting out. More and more people are coming up and saying, geez, I've been wanting to do this study with 500 samples, but nobody has 500 samples. Can we get access? There's an application form. We've tried to streamline this. This is not an NIH grant that's going to take you 6 months to prepare. You tell us the basics, what you want to do, have you done it before, is it a scientifically valid question? It's reviewed by the scientific advisory staff and the Accelerated Cure Project together. The Transverse Myelitis Association has a voice on that board with Sandy. We sort out all the paperwork, and then we distribute samples. It's very streamlined and quite user-friendly. And then the last thing's the key. This has never been done before. The data has to come back to the repository. So if I do a vitamin D study on everybody in the room, and then Jana does an Epstein-Barr study on everybody in the room, and it turns out that you have to have a certain Epstein-Barr and a certain vitamin D level, and that explains it, we only figure that out by having both sets of data back in the repository. So the data has to be returned. Never been done before. Why not? Here are some examples of studies that are going on, and you'll notice that all these involve TM, NMO, ADEM samples. We've released to a group I'm working with called Diagenics. This is where we're getting the blood-based test for predicting multiple sclerosis versus one-time events from. Beth Israel Deaconess in Harvard has a study on mitochondrial genetics. The Mayo Clinic is looking at mutations in aquaportin 4. Harvard just keeps coming back. They're looking at disease course based on genetic variants, UC Irvine, and so forth. So you'll see that this isn't just sitting in the freezer. We are getting active requests. The first publication citing Accelerated Cure Project samples has just come out. So the samples are being used and published upon. And it's things like this that are going to advance the common cause. So if we start getting back the mitochondrial data and the aquaportin 4 data, and maybe it's a combination of the two, we can start making insights into these disorders. So you can go to the website as listed here. You can check the protocol. You can look at our status. You can see how we're doing. If you want to enroll, there are instructions there. As of right now, you have to be at a site. We did this uniquely just to be able to augment our accrual. Outside of something like this, you need to be able to get to one of the sites. in order to enroll. And we're trying to streamline that as much as possible. So I'll turn this back over to Art.
I'm just going to very quickly wrap up and let you know some of the other things we have available in addition to doing the repository, what else we have that would be of use to you. All you have to remember about the next stuff I'm going to tell you is this link, acceleratedcure.org/offerings. And if you go to acceleratedcure.org, it's the first link on the page, and this will list everything I'm going to talk about. We have a bunch of information about MS. Most of this is MS specific, although this Working with MS book, even though it's called Working with MS, it's really working with any chronic illness. A woman who's one of our volunteers is a chronic illness coach. She has MS herself, and it's a really awesome book on helping you deal with career-related issues around having a chronic illness. And it's not one of these like high-level platitudes about, oh, you should have a good attitude. And she gives real concrete advice, specific things to say in certain situations. My wife has a couple blown-out discs, and she's used the book to help her interact with her employers much better and more successfully. So I highly recommend that. But there's a bunch of other educational materials. We have a sub-website on our website called msnews.acceleratedcure.org, and we every day post something about MS or TM or NMO or any of these diseases as we see it, whether it be research or just news, general news in the paper about people and so forth. And it gets kind of updated on a daily basis, and you can have the headlines or the bodies of the text mailed to you. It's a good way to keep up on what's happening. We have a free quarterly print newsletter. If you sign up, we'll mail it to you, and that just keeps you up to date on what we're doing, and we have various articles. They're not nearly the weighty tomes that Sandy sends out. They're 8 pages long, so it's just probably one session in the bathroom instead of a month. You can, the sign-up button on our webpage is right there. The new to us, try starting here. That's the one that takes you to the offerings page. And if you just click on the sign-up, you can get on our mailing list. Here's our contact information. We have commandeered one of the pharma tables out there that have left empty. So there's now, just outside that door, there's an accelerated cure table with a copy of the newsletter, a listing of the offerings, and a little brochure about the repository. So if any of you who weren't able to have your blood drawn here live near one of the centers or travel near one of those centers, grab one of those brochures, and it'll tell you how to get signed up. So, yes? No. Or not even the same place. Like my sister lives in New York. She went to the New York Center, and I went to the Massachusetts one. If they go to one of these sites, yeah.
Air trains direct to Baltimore from Seattle.
My question is mostly for anyone who's enrolled. Like if your diagnosis changes or if anything comes up, how do we just contact you, write you an email, and say, FYI, I now have this? So add that. Yeah.
So the question is if your diagnosis changes. So I don't know how it's going to work for the people who aren't near centers, but let me answer the easy version of that first. The people who are going to the dedicated centers, we follow up on a 1 to 2 year basis. We haven't done our first follow-up yet, but we're about to, at which point we will re-ask, has your diagnosis changed, and so forth. For those of you who are here, it may be kind of a one-time event. If your diagnosis changed, it would be good to email Ben or Jana and just let us know, and we can see what we can do with that.
But part of the contact information sheet you filled out is part of this prospectus. So you're going to get pinged in the future in some way for us to be able to change that data if we need to. Because you're right, that's valuable. The holy grail is, let's say we collected 10 samples today, and something about you changes in the future, we got the pre-change sample. Now we can really start understanding certain biologies. And so the longitudinal part of this is critically important. So it says in the consent form, if you decided to read it, there's a question. will you be contacted in the future? And it said, yes, you will be contacted in the future. You are under no obligations to respond, but we will hunt you down.
Well, it is a PDF on our website if you want to get it. All of our materials are on our website. We make everything available, except your personal information, of course. Yes.
I'm just wondering,
Yeah, I'm pointing at you. I was just making sure that I...
one of the reasons I didn't do this is because I'm Canadian and I would have to pay like $50 more to get my MRI thing. Is there a way for you guys to request that for my Canadian neurologist so that I don't have to cover that? Is there any way to do that?
Well, we can probably cover it if...
Doug Kerr will be seeing you in the back of the room at the end of this. He's happy to take care of all. No, we'll take care of it. Just let us know. We can help out.
Yeah, if you get people like that, just let us know. We can cut a check. Just a second. Let me get somebody over here. Paula. Well, the goal is, okay, we would add as many centers as we possibly could if we had the funding. So each center is, it's about a $60,000 a year proposition to have. So at this point, we're adding centers as we get funding. So for example, the Colorado Center is coming online later this year because the Tyler Hamilton Foundation is raising the money to start that center. So Tyler Hamilton, who's an Olympic bicyclist, has this foundation. He's running a big race, and he's going to raise $50,000, $60,000, and it's going dedicated to that. So as we go forward now, we're looking to raise the money to go with the site. So if anybody wants to help out with that, I know you'll all be stampeding up here afterwards, but yes.
I was declined from participating in the project because I had transverse myelitis 45 years ago, and I didn't have any documentation. But I did have MRIs in the recent years. What would be the difference in my MRIs now from when it was? I mean, it should show that I thought I had.
Yeah. So the question is, and I take full credit for any denial. So the question was, somebody was denied entry based on a diagnosis 45 years ago, and there were no MRIs 45 years ago, and we have more current and no medical records. And so case by case basis, I can sit down and talk to you and try and sort out. The honest answer is, as these samples go out, the value of the sample, and this is just the unfortunate reality, is directly tied to the clinical data we can document and the testing we can document about the individual as close to the time of diagnosis as possible. Here's the reason. Let's say it turns out that people who had a protein over 50 in their spinal fluid turn out 20 years later to have one thing. If we don't have just a basic amount of records, then it puts the repository at risk in terms of the samples. And I take full blame for this in terms of having to, on a case-by-case basis, make decisions. And I'm happy to sit down with you and see what we can sort out. The honest answer is, and we had this conversation this morning, how many people here have had their diagnosis change from its initial diagnosis to something else? Anyone willing? So I think we got about 10 to 20% this morning. Sometimes this happens between one assessment versus another. So when people come to our center to be evaluated for the repository, we do an assessment to make sure we agree with the diagnosis. And Jana, we're running a certain percent of disagreement, I think. I mean, she's laughing because she blames me, but that's the honest answer. And so we are limited to some degree. But I'm happy to talk to anyone who is denied to see if we can work it out. Right, so Sandy's bringing up the point, and it fleshes this out. A spinal stroke 45 years ago could have looked just like TM, but without certain testing, it got called one or the other, and vice versa. Someone got called TM, but it was actually a spinal stroke, or vice versa. So that's why we're trying to collect as much data around the actual event as possible so that we can look at it and say, it fits with an immune-mediated disorder. Because the moment I put the TM label on it, and the researcher asks for 50 TM samples, they're trusting that we have done our due diligence, that they meet the criteria of the disease. And so that's one of the issues. So before I forget, I know there's another question, but before I forget, those of you who left medical records that you need back, MRIs and medical records with the group, I've been looking at them. I'm going to continue looking at them today. They will be made available tomorrow during breakfast in the Alder Creek room. The team will be there to pass them back. If for some reason you need them before tomorrow morning, seek me out so I can put you at the top of the remaining stack to make sure I finish it and get it back to you today. But if you don't need it till tomorrow, great. They will all be available in the morning. And anyone who wants to do an all-night MRI session with me, room 308, we'll be putting up films all night long.
Okay, so I'd just like to, I'll be around to take additional questions. I don't want to, I can feel anxiety. But I do want to say one big thank you to Ben, Jana, and the whole crew at Johns Hopkins. We could not have done this project without them. They have bent over backwards. They have put in so much effort, unpaid, on their part to create this thing and get it going, and we're incredibly thankful for all the work they've done. And I'm also very thankful for Sandy for convincing us to add in these other diseases, because it really has made a big difference in how the studies that are getting done are taking place, because we now pressure each person who comes and says, oh, I want all these MS samples, and we say, and a whole bunch of transverse myelitis and NMO samples too, right? And they're like, yeah, okay. And so they're not averse to doing this research, they just never considered it. So by piggybacking all this work onto the MS research, a lot of studies are getting done, getting us data. Even if they're not directly analyzing it as a TM study, we're getting that data in place so somebody can do it. So thank you all for participating, and